In the previous video, we have understood that these Smad3 pathways that cause cell motility in a TGF-β induced cells. In this video, we will discuss what are the regions or domains of these Smad3 pathways that cause this cell motility in a TGF-β induced cells. So what they did is they take the Smad3 DNA, and in that they modified this MH1 and MH2 domain in a way that in one conditions they replace the N-terminal domains with the MH1 domain of the Smad1, and in other case what it did is they replace the C-terminal domain of the Smad3 or the with the MH2 domain of the Smad1, and those two things are been called as N1133 as they need only one mutations at 1133, and in the other case they did the mutations at N3311, and then check the effect on the cell motility in a TGF-β induced cells. So first, what I check the signaling pathways that get triggered in these different conditions. So for the control, they use the A549 cell lines. Here they can see that when the TGF-β is induced for 1 hour, they can see the phosphorylation of the Smad3, and in the case where a Smad3 has been knocked down, they do not see phosphorylations of the Smad3. But when the Smad3 has been infected using lentivirus, they can see the phosphorylations of the Smad3. And when this modified Smad3 is infected, that is N1133 or the MH1 domain integrated to the Smad3, and Smad3 is N-terminal side. In that case, we can see the phosphorylations of the Smad3, and similarly for this MH2 domain integrated with the Smad3 side, we can see the phosphorylations also. So in this case, we can see the phosphorylations signaling pathways get triggered, and thereafter they did the luciferase activity using the Kaga reporter, and they are able to see that luciferase activities are enhanced for all three cases when TGF-β is induced. When the cells are induced with TGF-β, the signals are higher for the wild type A549 cell lines. Thereafter, they check the different properties of the cell migrations and different properties of the cell motility. First, they check with the morphology. In the wild type cells, they can see the cells that are induced with TGF-β for 24 hours change its morphology. But for the this Smad3 knockout cells does not change the morphology. And here, when they infect the cells with N1133, in that case also they do not see much change in the morphology. But when they use these C-terminal domains or MH2 domain integrated with the Smad3, they see a change in the morphology. So from this, right now it looks like that the N-terminal or the C-terminal of the Smad1 plays an important role towards the change in the morphology. And thereafter, they also check the E-cadherin levels. E-cadherin levels also shows the same things, that is N3311 in the MH2 integrated with the MH2 of Smad1 integrated with the Smad3 shows a decrease in the E-cadherins, but the MH1 integrations does not show a change in the E-cadherins. And also using this the panel F also shows the same thing. There's so cell morphologies gets changes. Cell's morphology gets changes in the MH2 integrated domain in the Smad3, but not with the MH1 integrated domain in the Smad3. And when they check the E-cadherins using immunostaining, it shows the same results that we have seen in the Western blot using the panel E. That is, the E-cadherin levels goes down in the case of MH2 integration, but not in the case of MH1 integrations. And when we check the migrations assay shows that with the migration assay shows that when the A549 cell lines wild type is used, they see the more migrations, and when they infect this Smad3 knockout cells with the wild type Smad3, they also see the more migrations. But in the case where N1133 or N3311 is introduced, they do not see significant change in the migrations, indicating that for the migrations, this N1 and N2 domains of the this N-terminal and C-terminal domain of the Smad3 plays an important role. And after that, they check the using the uh, these Kaga proteins. They check the using the immunoprecipitations of the Kaga proteins. They check the uh, different types of different proteins that are involved in the SMAD3 pathways, that is phosphorylated SMAD3, and they are able to see the phosphorylated SMAD3s are present in the case of N3311 infected cell lines, but not in the case of N1133. So this from this also it comes out that that this MH2 domain of the SMAD1 plays an important role, and whereas this uh, N-terminal domain of SMAD3 plays an important role in the cell motility when the cells are induced with TGF-β. And however, this MH1 roles is not seems to be that much significant in the cell motility functions when the cells are induced with TGF-β. So far, they made this point that N-terminals of the SMAD3 and the C-terminal of the SMAD1 is essential for the cell motility in TGF-β induced cells. So I hope you understood how they come up to this final conclusions. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.